scientists decide to dive deep on one thing and spend their whole lives researching that one thing. And the fact that you help bring all these things together and sort of tell a story about what's going on in a complete soil food web and how we can fix this when things are missing.
If you don't have the whole food web present, you're going to lose species of plants. They're not going to be able to grow. And so what is the connection between this process being present and occurring in your soil versus not? Which of the plants are you losing? Which of the below ground organisms are you going to be losing? Which of the above ground?
Absolutely. And in fact, the next slide shows like the concept of what if we lost all of our protozoa in the soil food web? And that means that it would influence the bacterial community, since the
pretty rapidly.
Yeah. And maybe do things like what? Tie up a lot of nitrogen into the bacteria that then is not released for the plants.
Yep. The plants are never going to get that nutrient that's now stored in those bacteria. But of course, the fact that the bacteria aren't there to allow the protozoa aren't there to consume the bacteria, what effect is that going to have on nematodes or on microarthropods or ants or beetles, as well as the root system go backwards in this soil food web and start drawing all of the things that ultimately are going to be affected by the fact that the protozoa are no longer in the system.
Absolutely. It's like, we talk about like a cascade in ecology. When you remove something that's really key, I think about, so we're presenting for an African audience. What happens if you take lions or their top predators out of ecosystems? An overabundance of zebra or gazelles, all the grazing that those animals do unchecked now. I mean, the soil food web is just a tiny version of the big ecosystem.
Yep. And the same kinds of processes or functions are going on in the soil as above the soil.
Mm-hmm. Absolutely. So that brings us to, as we talk about wanting to restore something that's missing from the soil food web, and a lot of the strategies around that here at the school, where we teach people how to make really high quality biocomplete amendments, composts, teas, extracts. What would be some of the key targets then for the soil biology? What do we want to see in our amendments to bring back the soil food web?
All the diversity above ground of the plants. You've got to have all of the diversity of the microorganisms below ground.
Oh, absolutely. So, a lot of times I agree with you that people kind of come and they say like, well, what's the perfect little formula for the amount of fungi? And I'm like, you want a lot. You want a lot of different things in there, and you want a lot of fungi, because they're going to tell you something about, if they're thriving, it's going to tell you that your soil's got a lot of aerobic, habitat for them. It's going to tell you that you're not, going out of balance and getting something that's just devastating that population, like a disease or and some kind of antagonist. So yeah, it's not rocket science in a lot of ways.
We don't have to make it so difficult to try to figure out what's going on in soil. You don't have to know the 10,000 bacterial species that can be performing this function. So, we can just make certain we put maximum diversity, constantly be working to increase the diversity of the beneficial organisms. And one thing that we have to make certain is that when we're growing plants in soil, or we're growing plants in compost, 100% organic matter, when we're growing a plant in different conditions, that we understand exactly what the role and function of all of those bacteria need, or those bacteria want, and which are the ones that are going to be functioning this morning. because the
Mm,
temperature is going to get higher perhaps during the day. And now some of those bacteria that don't like things that warm will shut down and go to sleep. Somebody else gets to be king of the hill for a little while. And, then the wind picks up and it starts to cool things down, or you have a little bit of rain or you have a lot of rain. All of these factors are going to enter into who's reproducing as rapidly as possible. Are they doing their function to help your plant be as healthy and as happy as possible? Your plant has to have all of the nutrients that it requires. And what's this? Hey, plant, what's your secret? Well, how many of these kinds of nutrients do you have to have? And I'm afraid that we as human beings don't know that answer.
well, I mean, I've learned a lot from you about the. I keep coming back to like this sort of trust idea, like that I don't have to micromanage everything on a farm, but I can do a lot more of monitoring, seeing if something's really out of whack, and then trusting it if the biodiversity is there, And then, here's the other big thing that I've really picked up from you is the way that I thought about ecosystem succession before I came here was very plant community oriented, the way that the plants change, and then the way that you've linked that above ground ecosystem to what's happening below ground. Starting with, if we go all the way back to stage one, or maybe even we should call it stage zero, with a complete annihilation, a fire, a landslide, or something, and you have bare ground, what do you notice changing over time?
Yeah, you have catastrophic disturbances that are going to take these systems right back down to no nothing growing, nothing living, because the fire was 600 degrees Celsius, or the whole mountain slid down and buried your field under 40 feet worth of rubble. Um, Catastrophic disturbances.
Mm-hmm.
And so they tend to be bare because there's no nutrient cycling present at those early stages in succession. There's no organic matter in the soil. There no inoculum, perhaps even, of the microorganisms needed by the plants. And so I would really call that stage zero.
Mm-hmm.
Um, and so as soon as your area is visited by some birds, or a frog hops through, or you have voles and shrews and mice and all these other organisms that are zooming along, leaving behind an inoculum of
Mm-hmm.
all kinds of different microorganisms. The only ones that are going to survive and grow are the ones that have the enzymes to function
Mm-hmm.
under the conditions of all of that debris being left behind. Which of the organisms that we're looking at in the soil are going to be the species that we're going to need? We don't know.
Mm-hmm.
Let Mother Nature take part in the system of her design, and let the organisms that start to cycle nutrients once again, that start to build structure in the soil, holding water, holding nutrients, allowing air to move in and the CO2 out.
Mm-hmm.
Um, we need to establish all of those processes, and that's the first thing that nature does. So with enough time, you're going to get seeds in the system that can germinate under these conditions. And the microorganisms will then move to protect those root systems. And it's a definite protection game that these organisms are after, because those organisms need the energy from sunlight fixed by the plants.
Mm-hmm.
And then those sugars go down to the root system, and those sugars, the exact kinds of sugars that, and maybe some proteins, maybe some, there's a little bit of something in there from the nutrients that were in that seed, to allow the plant to put out the information of what the plant requires. When we're early in succession, you can see down at the bottom of this picture, that fungal to bacterial biomass ratio. So F for fungi, B for bacteria, fungal to bacterial biomass ratio. And when we're early in succession, there's almost no fungi starting to grow. These are not the right conditions for the fungal biomass to be doing its things, because fungal biomass wants very complex foods for it
Mhm.
to grow on. In competition with the bacteria are going to take anything that's simple. This is mine. Get your mitts off. And so it's very bacterial dominated because of the enzymes that and the way the bacteria use those enzymes means that this early stage of succession is going to be almost strictly bacteria.
Oh, I was just going to say, this is what strikes me every time when I hear you talk about this. Sorry to interrupt, but is that we've got a lot of places in the world where people have soils that are sort of like stage 1 and stage 2 soils biologically, and they're really trying to grow plants that are more for stage 3 or 4, a lot of our agricultural crops. And that's where we talk about, well, this is more like dirt
Yep,
way back here.
because there's not a lot of biology in there yet in the soil. Bacteria, it's usually just bacteria. That's all you find. You don't even find their predators most of the time. So how can you easily cycle nutrients? How do
Mm-hmm.
the plants get the nutrients out of the bacteria and what few fungi might be there? It's kind of laborious for them. And they certainly like the finally getting protozoa in here, finally getting maybe some bacterial feeding nematodes to start that system finally.
Going,
So,
yeah, going strong,
yep.
always moving forward, right? Unless something gets in the way,
Mm-hmm.
something's missing, some environmental factor, some human doing something like tillage, setting it back.
Yep. And people often, well, how much do you lose when you till the ground? Well, almost invariably, the data show that about 50%
Mhm.
of the microorganisms in the soil are going to be killed every time you till.
Mhm.
How many times a year are you tilling? And you can see where every time you till, you're going further back to that catastrophic disturbance. You're going backwards in that direction to the point where you're not going to be able to grow a plant.
Mhm.
And there are many places in the world where you can't grow plants in the dirt because there's nothing there for the microorganisms to live on, to grow on. Moss won't grow there. Algae won't grow there. So we just do things that are immensely stupid.
I think you've introduced a really great segue, which is saying, how do we jumpstart this?
Hmm.
Well, this is something that I've introduced already to the group, that regenerating soil biology is really something that we can do in about 4 steps. And we'll get deeper into especially step 4, which is really something that happens at every stage using the microscope. But it's understanding what's missing and then finding diverse local microorganisms, multiplying those on what we call biocomplete compost, giving them the conditions like the sweet spot, and then applying them to the soil so that we can, like you said, fast forward or jumpstart that process of succession.
Yep.
So let's dive into that.
Yeah.
Let's talk about composting principles. People want to know the nitty gritty. So, how do you start getting people's minds wrapped around how our compost is different than what other compost is?
Well, first of all, we're not allowing toxic materials to be made in our piles. Um, if you don't take care of your recipe and get your recipe right, and it goes anaerobic,
Mhm.
meaning, without oxygen or below a certain level of oxygen. So aerobic organisms want to have a oxygen concentration above 6 parts per million or 6 micrograms per milligram. So parts per million. Most of them need to have their enzymes. And let me get the exact explanation. Their enzymes, the enzymes of aerobic microorganisms cannot function below a part per million of 6 parts per million. It just won't work. So they stop growing. Somebody else is going to take over if it's anaerobic in that area. You're going to grow a whole different set of microorganisms, many of which will be disease-causing organisms. And when we have those disease-causing organisms growing and the plant is not getting the nutrients that it needs to be healthy, it's really easy for those pathogens to take over and wipe out your plants. Disease is going to take care of you. So we've got to have the compost made in carefully controlled conditions, or this whole process isn't going to work. We want to get these organisms back into the soil so that all the benefits we've been talking about that the biology gives you is present and functioning.
Absolutely. And we are lucky here that for the most part, a lot of those potentially problematic organisms are going to be pretty sensitive to heat.
Yep.
Because in addition to aerobic conditions, we're looking for particular temperature thresholds, controlling moisture, and then, I love the way that you explain time and turning. So what is it that you normally say? We have to make sure all material is hot enough, long enough.
Yep.
Yeah. I forget the exact. I mean, well, we don't. People can take our courses to get the exact information. There's a little plug for that, but
I'm just not quite sure what information you're asking about. You want?
Yeah. So if you're down lower, like 55C, you need that material to heat longer than if you're up near 70C. So that's the key principle I was going for.
Yeah, it's a time and temperature relationship. And so the hotter it is, the less time you have to be up at those temperatures, because the cell walls, the cell membranes, and the disease-causing organisms can't tolerate the length of time or the how hot something actually is. Both of those things will kill those disease causers. And it's one of the few times that I'm gonna say, let's get out there and kill it. Because we don't want the disease causing organisms. You can see that nature doesn't really take that attitude when she's trying to move things along in the successional process. She's going to make certain that the conditions in the soil choose the right sets of microorganisms to grow the plant that you want to grow. So you've got to be paying attention to that fungal to bacterial biomass ratio. But we're going to take these plant materials and we want to, as quickly as possible, wipe out the disease-causing organisms. We want to have this moisture. That means we've got to have a structure that's proper in that compost pile. So 50% of the water that's passing through that compost pile is actually retained in the compost itself. Oxygen, it's got to be above 6 parts per million, which means you've got to have airways and passageways. You've got to have freeways and, back, the up in the woods kind of thing where there's lots of curves and around and up and down it goes. But that allows oxygen to move into all parts of your compost. And we can keep that above 6 parts per million. Especially when we go in and turn it, we want to fluff that compost. We want to get everything well aerated again, and then let the microorganisms go back and get the temperature back up. We make certain that while we're turning, we're getting the oxygen in, and we're mixing everything. So we're getting a nice uniform compost that's being built. And moisture, oxygen, time, and turning have been monitored and we make sure that it's happened for long enough.
Absolutely. Well, there you go. I mean, the only other thing I'd love to point out to our audience today is that there's not a huge amount of compost here. Like the materials, maybe this is like a meter diameter, maybe a meter tall, something like that. And so we're not always just thinking, I think a lot of people go compost, it has nutrients. And we're going more like compost, we want it to be full of organisms. So you don't need as much because we have other ways of spreading those microorganisms, and we'll mention that briefly. But I mean, you said it, we're matching the plant needs, thinking about the fungal to bacterial ratio, where that plant is in succession. And then let's just mention briefly the recipe, because everybody wants the perfect recipe. But why is it that there is no perfect recipe?
Well, because we have different plants and we have different microorganisms and we have different environmental conditions that we've got to deal with. But in general, we can put together a recipe that is a good starter. This is where you want to start. And then you'll alter your compost process a little bit so that you make certain that you match up with the plant you want is going to get all of the nutrients. It's going to have all of the functions that come from the microorganisms. Your plant can make certain that the microorganisms do all the work.
Well, and that's the reason why we have whole classes dedicated to this, because people don't find that their first, second, or third compost pile really goes ahead and becomes biocomplete too often. It happens, but not that often. It's a learning experience, right? To get all of these factors right. And, I mean, I would just wanted to mention, again, this is what differentiates this from just any old compost, is in addition to organic matter, what we're really doing is we're improving the biodiversity of the soil food web. This is the source of a local population of organisms that we've then created the perfect habitat for the beneficial ones to grow, to outcompete any of the potential disease causers. And this is, I don't know, one of the big things, the aha moments that came to me here, because I studied soil biology and I was into fungi, or, an important thing that's missing from a lot of our farms. And I was like, but how do I help people bring them back? And then I came here and learned from you about biocomplete amendments.
We started destroying the biology in the soil, and we just made it worse and worse and worse. When we finally got to the point that we had destroyed enough of the biology in the soil, it didn't come back after you tilled. Then we started having to put on inorganic fertilizers, and then we realized that we were having horrible outbreaks of problem organisms. And so we learned to take toxic chemical materials and try to kill all of those disease-causing organisms. And that's never been successful. If it was really successful, you would only have to apply a pesticide once in your whole life. If some chemical actually killed all of that disease. But you know how this works. You kill off a lot of the disease, but it comes back at you two times worse the next year. And the next year it's worse. And the next year you have decided to go put something else in that field because you're not going to make a yield. And so we've got to understand that what's missing is the biology. We human beings made the choice to destroy those organisms because we believed they weren't important.
Right.
Back when I went around to all of the people in the departments at Colorado State University and asked them what was going on, and to a man, their response was, "Oh no, that's a horrible choice for a PhD, because you're not going to be able to be hired by anybody because the organisms in the soil don't do anything. They're not important." Can you imagine that? Given what we've discovered over the last 40, 50 years, it's really clear that the most important thing in soil are the organisms. It's
Absolutely.
really interesting to find how Mother Nature changes the plot crop that's going to grow over time. And so sooner or later, you're going to go through all of the steps and start growing old growth trees if that's what you choose to do.
Well, I think this will be our last slide then, just for this presentation, because we've already introduced thermophilic compost, but people are going to want to know that we can also make extracts and teas, and the value of those kinds of liquid amendments as well. And so I just wanted to put the picture of one of these brewers here, and and ask you, how did that kind of show up on the scene, after you were getting into composting and trying to restore the soil food web? How did you get into using liquid amendments?
Well, it's really hard to put out solid amendments. It's hard to make it even. It's hard to get the uh, solid material in underneath your plants. Um, you either have to put the material out over the whole entire acres that you're dealing with, and try to get as smooth an amount out. It's just not the easiest thing to do. And so, in general, what we actually do, unless the soil is just, well, if it's dirt,
Mm-hmm.
then we're going to have to use compost. That's, the first application is going to be solid compost because you don't have enough organic matter in the soil. We are aiming for a minimum of 3% to 5% organic matter. If you don't have that little increment of food, your microorganisms are just going to give up the ghost long before they're going to be able to do anything for you, long before your plant's in the system. So we'll have a solid, but let's say you've got the organic matter. So, okay, we've got the foods in your soil. Thank you for getting it in there. And now all you have to do is come along with a liquid application. So we take a certain amount of the compost and the certain amount, well, how much is that, Elaine? Well, it depends. Every time we um, have some unknowns in there. So we've got to look at what's in the soil. What are you missing? What's in your compost? Make certain that it's above minimum levels. It's above 135 micrograms per gram of bacteria.
Mm-hmm.
Same thing for fungi, 135 micrograms of fungi per gram. We want to make sure that you've got at least 10,000 protozoa, and we want the good guys. Thank you. You can have a ciliate sneak through there. 1 or 2 per sample is okay. Just no more than that. And then we want the good guy nematodes, the bacterial feeding nematodes, the fungal feeding nematodes, the predatory nematodes, but we don't want to see any root feeding nematodes. So we want to see that our organisms in your compost are above those minimum levels. So if they aren't above those minimum levels, then you may have to extract 1 or 2 pounds of your compost, check your compost tea. Excuse me, sorry. This is compost extract.
Mm-hmm.
And take a look using your microscope to discover, do I have the right number of all of these organisms or not?
This is great, Elaine. I think that between what you shared from your experience, and then also I'm going to be there in person to answer questions throughout the Innovations Club meetings. So this is a great place for us to stop on this topic, and then we'll get ready for the next video that we'll make together.